Hello, my name is Matt Irvin Arcillo, and my project is on Vibrio cholerae DNA stability test. Vibrio cholerae is a bacterium responsible for the disease cholera. It is an enteric disease, meaning it infects and colonizes the intestines. It spreads through contaminated water and can cause severe dehydration and diarrhea. If left untreated, within hours it can become fatal. About 140,000 people annually are reported to die every year out of the 4 million infected. However, since cholera mainly affects those in impoverished and developing countries, these numbers may be an underestimate. Cholera enters a person through contaminated food or water. Once it reaches the intestines, it releases a toxin called CTX. This toxin is responsible for the diarrhea seen in infections. Feces containing the pathogen can enter the water supply, where it can once again spread. Since cholera is mostly present in places where the water is unsanitized, the disease is very rarely found in developed countries. Cholera mainly affects countries without access to clean drinking water. On this map, it is shown that cholera is mainly present in sub-Saharan Africa and Southeast Asia. However, countries that have been recently hit by natural disasters, such as an earthquake in Cuba, make cholera reappear. In 2010, an earthquake in Cuba destroyed Cuba's ability to provide clean drinking water to its citizens. This caused a large cholera outbreak. After the earthquake in Cuba, there can be a large spike in cases seen in the Americas. However, excluding these instances, Africa remains to be the largest contributor to cholera cases worldwide. As of right now, the surveillance for cholera is insufficient. Surveillance can help us track cholera and its various serotypes, or strains, and understanding this can allow us to put into place better preventative measures. Surveillance can also allow us to give treatments to communities already affected, minimizing the global burden. A filter paper is a cheap material used to preserve a sample of Vibrio cholerae. Stool samples are placed on a filter paper and will be dehydrated, rendering the sample non-contagious. DNA is extracted via the Cholerae method, and it is very cheap and very quick. However, multiple types of filter papers can be explored in order to see which variety is cheaper to use and yields the most DNA. And this method is not limited to cholera. The extraction of DNA from filter papers using Cholerae is cheap and effective. It does not require special equipment and can be done within a few hours. Other methods, such as culturing, require special equipment and can be expensive and time-consuming. The goal of my research is to see which type of filter paper will be able to produce the highest concentration of DNA consistently over a time span of 6 months. We will be comparing a Whatman 903, a Whatman W1, a Fisher P8, a rapid diagnostic test, or RDT, and a Scott paper towel. I receive each sample pre-prepared. Each filter paper will have a sample of cholera culture spiked onto it, and data will be collected through various time points within the 6-month period. DNA concentration will be measured using a nanodrop. A nanodrop uses spectrophotometry, which passes a beam of light through the sample to detect the levels of DNA within it. Each sample will then be confirmed using a PCR assay, which detects the presence of the cholera toxin and a cholera protein in our samples. This is my data graphed over a 6-month period. The rapid diagnostic test, or RDT, consistently produces a high level of DNA concentration and tends to have the highest concentration out of all the samples. Next to it would be the Whatman 903 and the Whatman W1. The paper towel produced a consistent amount of DNA nearly every time it was measured, yet it produced the least amount of DNA almost every time. Here is each of the samples graphed over a 6-month period. Each sample had 3 replicates when measured every time, except for the RDT. The vertical lines represent the ranges of each sample measurement. This is the average range of DNA concentration for every measurement for each filter paper type. While the Whatman 903 consistently produced a high level of DNA every measurement, it also had the highest range, which implies that it is not as consistent as we thought. The paper towel, while the most consistent, did produce the least amount of DNA every time. Data analysis. The 903 and RDT consistently produced the highest concentration of DNA. However, the 903 did have a high replicate variability. Paper towels, while producing consistent levels of DNA, produced a low level of DNA, and this can be seen as not all paper towel samples were verified using PCR, and this implies that we're not even sure cholera DNA was present in each sample. Before beginning my experiment, I believed that the Whatman 903 cards would be the most reliable and would yield the highest concentrations of DNA nearly every time. While my data did show that they were able to yield the highest concentrations nearly every time, excluding the RDT, they did have the highest variability for each sample replicate. Also, before my experiment, I believed that the concentration of DNA would decrease over time. This was not present in my data, which did surprise me. This implies that the DNA decay in filter papers does not happen as fast as I thought it would be. This also means that the stability of DNA on filter papers is much more reliable than previously thought. One thing that did surprise me though was that some paper towels were able to produce a level of DNA, and while not every single paper towel was able to be verified using PCR, this implies that they can be a viable alternative to the regular type of filter paper used in our labs today. Paper towels are much cheaper than a Whatman 903, which means that if we were able to find a more reliable type of filter paper, we could be able to use them in our labs. In future research, I would like to see molecular analysis of each filter paper. If we could compare what makes each filter paper as effective as it is, and perhaps create a filter paper that combines all these features, such as the cheapness of a paper towel and the effectivity of an RDT or a Whatman 903, then we could create a filter paper that would be optimized for DNA extraction and preservation. In future research, I would also like to test with fecal matter instead of cholera culture in order to see if the DNA I produced would be the same. I would like to thank the CIR lab and the Enterix lab at the Johns Hopkins Bloomberg School of Public Health for assisting in my research. Thank you, that is all.